All right, thank you again for joining in today. Next slide, please. So today's coffee seminar is from cell to ecosystem, understanding environmental contamination by Professor Catherine Propper. Before I will introduce you, next slide, please. Some of you might not know me. My name is Sudhir Yadav. I'm Senior Principal Research Fellow at Center of Crop Science in coffee. And as normally we do, before we start, we would like to acknowledge the country. Next slide, please. On behalf of everybody and University of Queensland, I would like to acknowledge the traditional owners and their custodianship of the land on which we meet today. We pay our respect to their ancestors and their descendants who continue cultural and spiritual connection to country. We recognize their valuable contribution to Australian and global society. Next, please. So some housekeeping things before we start the seminar. As usual, the seminar is scheduled for 12 until 1 p.m. At the end of the seminar, there will be opportunity for asking your questions to Professor Propper. Please take note that use Q&A function or Q&A button to put your questions, not in the chat button, and we will read your question at the end and address them at the end of the seminar. Next slide, please. So it's my great honor to introduce Professor Catherine Propper. Professor Propper is engaged with the Department of Biological Science at Northern Arizona University. She is an environmental endocrinologist and ecotoxicologist and serves as the lead for National Institute of Health funded Southwest Health Equities Research Collaborative. Her research focus is mainly on environmental contaminant and how that affect development, reproduction, and behavior. As one would expect, she's a well-renowned international scientist and has published a number of articles in her research domain. She has served on a number of panels and scientific advisories related to water quality and environmental toxicology, including with the environmental protection agencies. We have been fortunate to collaborate with Professor Propper for long in my current as well as previous role. And at this time, we were able to have her here as part of Fulbright specialist program to come here and interact with coffee colleagues. With that, Professor Propper, I will hand over to you to talk us through your work and information you would like to share. Thank you very much again.
Well, thank you very much, Sudhir. I really appreciate both the invitation to be here today and the invitation for the whole visit. I too would like to acknowledge the traditional owners and their custodianship of the lands on which we meet today and pay my respect to their ancestors and descendants. Also, though, I would like to acknowledge that Northern Arizona University sits at the base of the beautiful San Francisco Peaks on homeland sacred to Americans throughout the region. We honor their past, present, and future generations who've lived here for a millennia or lived there for a millennia, and will forever call that area on which I get to live home. With that, I'll begin my seminar. I'll be talking about from molecules to ecosystems and the complexity of understanding environmental contamination. I want to acknowledge both Coffee and the Fulbright program and my institution for supporting this trip. All right, first I'd like to give a little bit more of a land acknowledgement and who I am. I grew up in Los Angeles, California during the 60s and 70s. You might be able to hear in my voice that I lived in the smog. I think that got me first interested in studying the impacts of pollution on wildlife and human health. I ultimately moved to the beautiful Flagstaff, Arizona, and Northern Arizona University, which sits on sacred lands underneath these remarkable old volcanic peaks. How did I get here to Australia? Well, it was kind of a long pathway. About 15 years ago, I met Dr. Grant Singleton, who was working then at the International Rice Research Institute. We talked about a project looking at amphibians as biomonitors for pesticides in the rice agroecosystem, and I'll talk about that more in a minute. Grant introduced me to Professor Yadav, who was then working with a graduate student, Nishanka Jayasiri, in Sri Lanka, and we did a project that utilized the ecotox knowledge base from the US EPA as part of her project. Sudhir thought that it might be a good idea to do a full-blown workshop on that. He got together with Barbara George Jagali here at Coffee, and together we worked with the Fulbright Specialist Program to ultimately land me on an airplane across the Pacific. All right, so what work do I do in my laboratory? Well, I study model species to understand the effects of chemical contamination that is in the environment. I look at amphibians and fish largely, because it allows us to evaluate aquatic exposure. And aquatic exposure acts as a really nice surrogate for the in utero environment. I'm going to take a sip here. The other really important fact to know is that the hormones that regulate all of their physiological systems are the same, or chemically and molecularly nearly so, and have similar actions because of the conservative nature of the endocrine system throughout evolution. We also are now beginning to use aquatic invertebrates to allow us to evaluate population and ecosystem effects, but I won't be talking much about that today. Another part of my job is training programs aimed at first generation and other historically excluded groups in the United States. I've had funding from the U.S. National Institutes of Health, National Science Foundation, and the Alfred P. Sloan Foundation in support of this project. It includes undergraduates, postdocs, MS, and hopefully PhD students. We've worked with faculty training as well. And I've had very interesting conversations while here about trying to develop an Indigenous or First Nations exchange program. But let's go back to endocrine disruption and chemicals in the environment. A little bit of a brief history comes from, most of you have probably heard of Silent Spring and Rachel Carson. She started noting that there was a lot of population declines of birds and found that it looked like it might be caused by DDT, one of the most commonly used pesticides in the world. Still, in some places of the world, it's the best way for mosquito control. She published Silent Spring, and then about a decade later, we found that DDT actually binds to the estrogen receptor. And while that might not be the cause of eggshell thinning, it could be the cause of a number of other reproductive abnormalities. Next came Theo Colburn in the early 90s. She started gathering all this data about birds and population declines around the Great Lakes. She got concerned as these papers were demonstrating a lot of reproductive and developmental abnormalities. She helped organize the Wing Spread Conference in July of 1991, brought in a lot of wildlife and human endocrinologists to take a look at the problem. They published this in 1992. She went on to publish a lay version of this called Our Stolen Future in 1997 that got the attention of the general public and of the US EPA. All right. So when I'm thinking about my research, I always end up going back to the rhyme of the ancient mariner with water, water everywhere, and all the boards did shrink. Water, water everywhere, nor any drop to drink. And I like to use this analogy because ultimately the mariner killed the albatross that was protecting the ship. And the albatross was hung around his neck, and it became an analogy for the fact that they were stuck at sea with no water to drink. But of course, now all of our water systems, there's no control sites we can use for any of our studies. We call them reference sites. Water everywhere has chemicals from human use in the water systems. It's causing now, of course, we know there's plastic pollution everywhere. There's pharmaceutical pollution everywhere. Pesticides are getting into water systems. There's wildfire ash and the flame retardants used on it. There's biochar, and we're using a lot of biosolids on our agricultural systems. In fact, Sudhir and I have been talking about collaborating on this part. And then the European, US, and some of the Australian systems are now trying to develop these new approach methodologies to address this problem. And of course, we've got the elephant in the room, which are the forever chemicals, or PFASs, polyperfluorinated alkyl substances, which we're now studying in my laboratory as well. All right, so I always like to go back to what is the endocrine system. Not everybody is an endocrine physiologist, though I consider the endocrine system, like everybody considers their system, the most important. It starts up in the brain in the hypothalamus. It goes to the pituitary gland. The hormones are released from the pituitary that affect both other endocrine processes and release of hormones that then affect everything to do with your physiology. So then what are environmental endocrine disruptors? They've been called those chemicals of emerging concern. They get renamed every few years. But the EPA definition is they're an exogenous agent, or it's something that comes from outside that interferes with the synthesis, secretion, transport, binding, action, elimination of natural hormones in the body, which are responsible for maintenance of homeostasis, including reproduction, development, and behavior. Basically, they're anything from the outside that impacts endocrine function and hormones. I've given some examples here. On the left is endosulfan. Endosulfan was a pesticide. It's now banned. It was one of the first ones I started studying in the mid-90s. DDT, of course, in the bottom. And then even ethinyl estradiol, used in birth control pills, has endocrine disrupting functions when wildlife are exposed through water systems. There's many classes of these chemicals, of course: pesticides, industrial chemicals, phytoestrogens, metals. I'll talk a little bit about arsenic. Pharmaceuticals. Many of you probably know people that are on tamoxifen or a similar drug for breast cancer. Ethinyl estradiol, used in birth control pills. And I want to pause here and just say, I am not against use of these chemicals for agriculture for the appropriate uses. There are appropriate uses for these. We certainly aren't going to be asking people to not use pesticides if their agricultural crops are at risk. We need to feed a lot of people. I'm never going to ask women to go off birth control because of the long-term impacts. We need to find better solutions. Ash and fire retardants, biosolids, and then the complex mix that often ends up in our water systems through biosolids and through wastewater. The chemicals in there are often at concentrations that we now are finding within our bodies, which indicates that there may be real problems that we see. If we're seeing it in wildlife, we may be seeing it in us too. All right, there's the importance of timing of exposure. That's really key. If you're exposed during key developmental time points, it can interfere with key processes of development, and the outcomes of that kind of disruption is often permanent. As adults, disruption occurs. Exposure can disrupt appropriate ongoing endocrine function, and sometimes those effects are permanent, and sometimes they can be reversed once the chemicals are removed. One of the scarier parts, though, is that several studies now suggest through epigenetic processes that exposure of one generation can have transgenerational effects impacting developmental processes in subsequent generations. All right, and I've kind of gone through this. There's multiple sources of pollution, And in fact, what we're finding is that low doses of these compounds can actually have effects. So when I'm talking about a parts per billion concentration, it sounds very small, but the endocrine systems evolved to be very sensitive. All right, so I'm going to start with bullfrogs here. Bullfrogs are a nice model. The bullfrogs are big. The tadpoles are about that big, which makes it really easy for us to see what's going on inside of them and to look at their tissues. Estradiol in a frog looks like this. Estradiol in a human looks like this. They're the same chemical. It's the basic form of estrogen that affects development and reproduction in both frogs and humans. I want to talk a little bit about how do we get an ovary or a testes. And ovaries and testes start as bipotential tissues. That means it's the same exact tissue. What genes turn on determine whether it becomes an ovary, which you see in the middle panel here in a frog, or a testis, which you see on the right panel. Those hormones that end up being turned on include estrogen, if you're going in a female ovarian direction, or testosterone in a male direction. In frogs, the gonads start undifferentiated in that very early tadpole stage. By the time the tadpoles have a hind leg with 5 little toes on it, they have differentiated into either that ovarian or testicular tissue, making it really easy for us to see how gonadal differentiation has occurred under the conditions of exposure regimes for chemicals in the environment. So we evaluated the sex ratios and gene expression for these genes involved in this gonadal differentiation problem or development. And this work was by Stephanie Wolf in my laboratory. This shows you a tadpole. We took tadpoles from 2 different areas, J.D. Dam Lake, which is out in the middle of nowhere, about an hour south of Flagstaff in the forest. It's our reference site. Again, we cannot call it a control really, but it's going to have many fewer chemicals in it, versus the Rio de Flag, which is a little creek that runs through Flagstaff that completely runs on wastewater effluent, reclaimed wastewater effluent, coming out of our Rio de Flag treatment facility for wastewater. All right, so I'm just going to show you the results here. Tadpoles from the Rio de Flag had a lower proportion of males. If you take a look at the top panel, you have the distribution of females and males on the y-axis, and then J.D. Dam, the cleaner site, is the first bar, and the Rio de Flag, the one receiving wastewater effluents, on the right. And we had a significant decrease in the number of males coming out of the Rio de Flag. And this experiment was repeated a couple times through different graduate students. We then looked at expression of a gene that leads to an enzyme that converts testosterone to estrogen. You don't get estrogen without going through testosterone. So even in humans, all the estrogen we produce goes through testosterone first. So if you look at the expression of this gene overall between J.D. Dam and the Rio de Flag, the wastewater facility, it was elevated, suggest in the Rio de Flag and the wastewater, tadpoles from wastewater. That suggests that there was probably more estrogen being secreted during development, which might explain our sex differentiation issues seen in the top panel. All right, I'm going to shift over now to thyroid hormone disruption. Thyroid hormone is really critical during development. If you don't have appropriate thyroid hormone secretion during critical phases of development, which is in the perinatal period in people, and during the transition from tadpole to frog in amphibians, you get a lot of problems. And again, thyroid hormones in human is similar to thyroid hormones in frogs. So going from a tadpole over to a frog, again, is completely thyroid hormone dependent. If you don't have enough or sufficient thyroid hormone secretion, all you get is a bigger tadpole. So The morphological endpoints for going from tadpole to frog are easy. You have hind legs popping out, then forelimbs, and the tail going away. So these give you morphological endpoints. And this was really the basis for the US EPA's original amphibian metamorphic assay that then went into 2 different tiers for evaluation of chemicals in the environment for being thyroid hormone disrupting. All right, so this was our first experiment, was we wanted to look at the impact of exposure to wastewater as an indicator for complex mix effects on thyroid hormone dependent development. Of course, all of us are exposed to not just one chemical at a time, but to these complex mixes. So we asked, does exposure to wastewater affect this developmental timing? We had tadpoles. We used African clawed frogs as a model. We put them into 2 different types of controls. PTAX is the standard media used to raise these tadpoles from the amphibian metamorphic assay. And then we also used RO water on the right that we salt matched to the wastewater that was coming out of the Riota flag. And we put animals in each of these different kinds of treatments and then just looked, how long does it take for them to go from tadpole to frog? The results are shown here. On the y-axis, we have mean stage. This is just a staging system. If you get to stage 66, you're now a frog. Below that, you're still a tadpole. On the y-axis was the number of days the animals were exposed, and you can see that, let's see if I can get my little pointer up. You can see here that the guys that were in the wastewater accelerated their metamorphosis compared to either of the controls. Then we did an experiment where we wanted to look at wastewater extract and see if it impacted expression of thyroid hormone sensitive genes. So of course, when you give animals thyroid hormone, it causes a lot of downstream effects for how your genes are expressed and converted to proteins that lead to all the developmental processes. So we had animals not exposed to thyroid hormone and then exposed to control water, a 50% concentration of wastewater and 100% concentration of wastewater. And then we also added thyroid hormone and did the same because we wanted to make sure that we weren't going to see inhibition of thyroid hormone dependent processes as well. Now, we predicted that we would see acceleration based on the previous results that I just showed. All right, I know this is a little bit of a complicated slide. I'm going to walk you through it. Here we have animals, it's always getting this pointer is a little tricky. Animals not exposed to any wastewater. And you can see thyroid hormone, exposure to thyroid hormone, increased expression of 3 of these 4 genes that are thyroid hormone sensitive. If you then take a look at what happened when we gave them the wastewater, you can see that there was an even greater response to these thyroid hormone sensitive genes in all cases, which could mean that these results suggest that our acceleration of metamorphosis that we saw earlier is because something in the wastewater is accelerating these thyroid hormone dependent processes. All right, we've also looked at heavy metals, and heavy metals are a natural form of environmental contamination. They also end up becoming concentrated under conditions of mining. I have different heavy metals here. This is a mine. We have the Gold King mine spill in Colorado, which contaminated a good part of one of the rivers feeding the Colorado. And on Navajo, water is life, and there's been a lot of uranium contamination. All right, we wanted to look at arsenic disruption of androgen-dependent processes. Testosterone is an androgen, and this is the work of Riley Smith. In this case, we used a different model. These are mosquito fish. I believe you have mosquito fish here in Australia too. Mosquito fish, males have this elongated anal fin, females have this fan-shaped anal fin. Getting this elongated anal fin happens during development, and it's an androgen-dependent or testosterone-dependent trait. We put these animals in increasing concentrations of arsenic that were environmentally relevant and then looked at development of this anal fin as well as overall body shape. All right, so this table shows you the results. Here we have the doses on the top. So this will be read across from left to right. The doses increase from high parts per trillion to just below the drinking water standard in the US, and to concentrations that we often find in water systems naturally in Arizona. We looked at the condition factor of the fish. We looked at the liver function of the fish, because a lot of detoxification happens in livers. We found that the condition factor in these juveniles that we exposed to these concentrations wasn't impacted, but the liver was more than double in size. We then looked at reproductive indices. GSI is the gonadosomatic index. It's just how much the testes weighed in comparison to the body. And we found no effect here in the juveniles when they were exposed. But when we looked at what was happening to the length of their anal fin, that's called a gonopod, compared to their body length, we saw a dramatic decrease in the length of that fin, even at levels below the US EPA's drinking water limit. We then also looked at their body shape overall, and we found, again, changes in body shape. This is a non-parametric, it's like a non-parametric PCA, and found that there was a pretty dramatic change in the shape. And this was particularly predominant in the muscle that controls that anal fin's movement for fertilizing the females. We did this, I want to just say, we also repeated this study a little bit differently by taking fish out of water systems in Arizona with different arsenic concentrations. These were adult fish. By then, their liver was looking like it was giving out, and the gonadosomatic index, that is, their testes, were smaller in areas from higher arsenic concentrations, as well as their gonopod, the anal fin, was shorter. So our results in the lab parallel labs and the results in the field. I want to talk a little bit about pesticides in agriculture and the work we did at the International Rice Research Institute. And all of you involved in agriculture, of course, know rice is a food staple for many people in the world. It's very pesticide intensive, as are much of our agricultural systems. And these pesticides may affect wildlife and human health through overt toxicity, but also through endocrine disruption. We looked at butachlor, which is a commonly used pesticide throughout Southeast Asia on rice. We did this experiment in the greenhouses at Erie. And we looked at whether or not butachlor, again, impacted thyroid hormone dependent processes. So we're back to thyroid gland. Again, I always ask people, feel your thyroid gland, because it's such an important organ for how you feel. If you take a section out of the thyroid gland and look at it under the scope, you see these cells that make a precursor to thyroid hormone. Put that into the middle here, where it's then converted to the active hormone. We used your favorite animal here in Australia, cane toads, to be the model system for this experiment. Cane toads are also introduced in the Philippines and are considered a pest species. We took a look at their thyroid gland during development, and were able to measure this thyroid gland and look at it to determine whether or not butachlor was having an impact during development. Molly came up with a very clever experiment. I'm going to walk you through it slowly. She used a formulation of butachlor. She exposed animals for 12 days, but some of those animals were exposed only as eggs and hatchlings and then put back into clean water. Some were exposed as tadpoles, but not as eggs or hatchlings. And then some were exposed continuously. And then this little graphic shows what happened to their bodies, their developmental processes. You can see development was generally slowed no matter when they were exposed. And the highest concentration, this is 200 parts per billion, all of these are environmentally relevant, killed the frogs. We then looked at their thyroid gland. So again, let's read across here. The control panels are here. The lowest dose, this is 2 parts per billion, 20 parts per billion, 100 parts per billion. You can take a look here at the glands. You can see the glands pretty much look okay, no matter what the control was. If you go to the 2 parts per billion, those exposed as hatchlings, well, it looks okay. During tadpole, well, maybe not so good. And by the time they're continuously exposed, even at 2 parts per billion, those thyroid glands look pretty sad. The same thing for 20, and at the highest dose, we don't even have tadpole follicles because we have no tadpoles. We then did an RNA-seq study on the intestines, looking at every single gene that was expressed differentially between the controls and the exposed animals. And this heat map represents 1,000 genes going down the heat map. These are each of the individual animals. This is the control group. Clearly, you can see there's a difference in expression of the genes between the control group and everybody receiving butachlor. Some of the more scary data I've seen. And a lot of these genes are involved, of course, in development, metamorphosis, and metabolism. All right, the last study I'm going to talk about today is one that was done by Nishanka Jayasiri. She was Sudhir's graduate student. She did a PhD that was pretty spectacular, using a case study in one of the watersheds within Sri Lanka. So here's Sri Lanka down here. I just lost my pointer. There we go. Here's Sri Lanka. Here's the island. Here's the watershed she studied. And she looked at 3 different points along the watershed, interviewed farmers about their use of pesticides, what pesticides they were using, and then measured the concentrations of the pesticides at each site multiple times throughout a growing season. And then we took a look at those concentrations and used the Ecotox knowledge base that the US EPA has put together to evaluate whether or not those levels could be potentially harmful to the ecosystem and the organisms living in those water resources. So first, she collected this pesticide application rate from farmers. She quantified the amount of pesticide use above the recommended levels, standardized to 1 for each pesticide across all the farms, and found that most farms exceeded the recommended application rate. So here you see the recommended application rate standardized to 1. And here for each of these pesticides, they were using many of them above the recommended use rate. She then used Cornell's environmental impact quotient calculator to determine overall risk across all the pesticides for the 3 sites within this watershed for consumers, workers, and ecology. This quotient is from a calculator developed by Cornell. And you can see that even for consumers and workers, there's some environmental risk of exposure. And then under the ecology side of it, it was even higher for organisms living in the environment. The maximum detected concentration for some of these pesticides ranged from 16 parts per billion. Remember, we were seeing effects at 20 parts per billion and even 2 parts per billion on butachlor, up to these parts per million levels for some of the pesticides. And she determined the potential risk outcomes based on available data, like I said, from the Ecotox knowledge base. All of the pesticides evaluated through this knowledge base demonstrated potential risk of toxicity to species, non-target species, within the levels found in the watershed. And some of these effects provide, some of these species do provide ecosystem services back to the farmers. So for example, spiders that were affected can have pest control services. And of course, fish can have many different kinds of ecosystem services back to the farmers, including pest control, food, and economics, as well as on the flip side of that, leading to further contamination if they're used as a food crop. All right, some of the other work in my lab is dealing with at the ecosystem level. I won't talk too much about it, but because of climate change, there's a lot of fires that lead to smoke and ash contamination and fire retardant use. The picture on the left is from the second of 3 times I've had to evacuate my house from fire. This was taken from my driveway as we were packing up the car. And then, of course, there's a lot of fire retardant use. This picture was taken from a fire retardant drop on the Palisades fire in Los Angeles, where my folks also had to pack up their car to be ready to evacuate. And so this is something that's really near and dear to my heart. Context is very important. Temperature and contaminants interact with each other. We're now doing a study in this beautiful animal. This is a tiger salamander, again, completely thyroid hormone dependent on their development. I haven't talked about stress hormones, but stress hormones also are important in metabolism. They're called stress hormones, but they affect a lot of different things. And they're also important to this process of metamorphosis. And of course, in humans in development, preemie babies are often given cortisone or cortisol-like analog to get their lungs to develop properly. Species respond differently to the same exposure. So of course you need to consider a frog isn't a human, and even not all frogs are alike, not all fish are alike. There's some differences in how they metabolize or work with those hormones in their environment, and then the chemicals. Social interactions can have an influence. There's developmental exposures can influence outcomes, possibly through these epigenetic mechanisms that can transfer through to the next generation. Past evolutionary history can lead to population level differences in outcomes as a result of selection. So if animals are living in multiple generations in some of these contaminated sites, they do evolve some processes for dealing with that contamination. And that also means you have to be careful in what population you use if you want to determine whether unexposed populations may be sensitive to exposure. And then, of course, nobody lives in an environment where you're only having one exposure. Everything has exposure to multiple chemicals. And in fact, when you look at some of the databases that are available, so for example, this database from the Centers of Disease Control, we do a national report on human exposure every year, every 3 years. And you can see that the levels in people's plasma and urine is the same basically as what we're seeing coming out of wastewater treatment facilities. We have a number of these different databases available in the US. There's the endocrine disruptor screening program, and there's a dashboard for that. We've got the Ecotox dashboard shown here, and then the CompTox dashboard. And these were the ones I gave the workshop on 2 weeks ago when I first got here. Seems so recently. All right, I'm going to talk about all of this under the context of a One Health perspective. And this is a figure I took out of the USGS website. That's a geological survey, talking about the intersection between animal health, human health, and ecosystem conditions associated with chemicals in the environment. But of course, this intersection goes for all different kinds of environmental interactions between animal and people. It's not just all about animals involved in agricultural issues and zoonotics, though those are critically important, as it is for wildlife-human interactions, but also with these chemicals in the environment and how they interact through food webs and water systems. And last, I really want to thank both Sudhir, Barb, and all the people that worked so hard to bring me here, both here and through the Fulbright program. All of my collaborators, these are just the folks who collaborated with me on some of these projects. And then also my graduate students here who worked on this project. And they also helped mentor a slew of undergraduates who worked on this project. With that, I will put it back to questions to Professor Yadav, and we can go from there. Thank
Thank
you
you
so
very
much.
much. Thank you very much, Professor Proper. I think this was fantastic. One thing which I particularly like is sort of landscape of various species as well as geography that where you have worked, which is basically making the conclusion very, very solid that often people say that, hi, this is the result you got in your locality. But I think you are making a very strong point that this is a global issue, and irrespective of where we are, we can see those spillover impact. The another very interesting thing which you very strongly argued and presented is this interlinked, sort of taking toward the final slide where very nicely you mentioned about the linkage, or looking it with the food lens, and thinking about one health system, which many times, being in focus with the agriculture sector, we focus on one domain and we are not able to see. So that was fantastic. I can already see there are questions that started to come. While I will again remind everybody that please put your question in Q&A box, not in the chat one, and we will go through them shortly. While more questions are coming, perhaps I would like to get a little bit more thoughts from your side, Professor Proper. Specifically, I mean, you showed us beautiful pictures of amphibian fish. Those are fantastic, and I think you made very, very clear through your results that there are direct correlation with human health. But want to get your insight that do we need to still be a bit cautious about when we translate results from amphibian or fish to human health? Are there some gray area where we need to be mindful?
Thank you for that question. It's really an outstanding question, Sudhir. And of course, we do. There's a lot of differences in the pharmacokinetics of how some of these receptors and enzymes function that can determine things like shifts in the dose responses that cause these effects. There's ways of decontamination that our body deals with. And we often see, even in our studies, when we're looking across many genes and expression of many genes, some of those decontamination genes get turned on early to help with getting rid of those chemicals out of our system. And then also we do need to be careful looking at humans. And of course, there's a lot of studies in rodents and sometimes other mammals that also are showing actually similar results. The dosing regime is a little more complicated because it has to go through food or water. So it's not as simple, and you end up with much smaller sample sizes often because it's more expensive just to use those animals. And currently across the globe, right, we're trying to reduce the use of especially mammals as models for humans. And then secondarily, the way we can start looking at this more is through epidemiological approaches. And there are a number of epidemiological studies showing disruption or showing correlations between exposure risk and exposures and endocrine function problems. Those, of course, are correlations. And correlation doesn't mean causation, but we need to use these animal models combined with in vitro models, meaning cell tissue models, combined with, which is what the CompTox database uses, right? Combined with these broad literature databases like the Ecotox knowledge base in order to make those connections between correlation and causation. Of course, we can't do these experiments in humans for obvious reasons. And so it's you can't say that there's a direct effect that at this dose humans are going to be impacted, but you can look at what's in human plasma both during development and in adults, and then look back into the literature in animal models and in tissue culture models and take a look and see whether or not those correlations are really suggesting a causative impact. That was kind of a long answer to your question, but there you
No,
go.
I think that was good. Fantastic. We already started to get number of questions, so we'll try to address as many as we can. First question is from Harvey Santos. Harvey is a researcher in coffee, recently passed out. He completed his PhD. Congratulations, Harvey. So he appreciates your talk, and he's asking that given that thyroid hormones are so critical for timing and tissue remodeling, what do you see as most informative endpoints for detecting early thyroid disruption in wildlife? And the second part, and how well do these amphibian model help us predict similar risk in other vertebrates, including human?
Yes, and also a very outstanding question. I've been working a little bit with very simple predictive models by pulling in data from the Ecotox knowledge base, just based on overt toxicity, which is often linked to, it's called lethal doses of 20%, 50%. How much does it take for animals to die? I don't like that as a model for endocrine disruption or development. I think it's a very gross endpoint. But so I guess to the first part of the question, I would say most important, I would say if you're disrupting development through thyroid hormone developmental processes and you're not going to even get a functional adult at the end, that thyroid disruption is really important. As probably many of you know, there's a lot of people now that are having thyroid hormone dependent processes with their metabolism. I think, so that's primary. Secondary, I would say if you could start messing with reproduction, you start getting a lot of problems in humans. That would be linked to fertility in wildlife, and in domestic animals, that starts leading to problems not only with fertility, but with populations and ecosystem processes. And so in that regard, it becomes a systems level approach, which Sudhir and I have been talking about a lot. And let's see, so now I'm trying to remember the second part of the question because I had an answer to it. Can you remind me the second part of the question, Sudhir?
The second part of the question is, and how well do these amphibian model help us predict similar risk in other vertebrates,
In
including?
other vertebrates. So, and I've been playing a bit with some modeling on that. I work with some colleagues at ASU, and we've been working a lot with heavy metals coming out of mine systems and using that ecotox knowledge base to figure out where those concentrations might be problematic in water systems in Arizona. And from that work, we decided to take a look at some of the potential for developing predictive models. And we find that at least we're starting with LD50s, 50% lethal doses, because that's the simplest model. We're finding that they are pretty predictive if you can look across multiple organisms. And then if you have an organism and you don't know that say the lowest effect level you could go to before you cause death and destruction, you can use that LD50 as a predictor for where it might be a low effect or no effect. And so, but those are going to involve predictive models that I'll probably pull in Professor Yadav for, and maybe some of his students to help me develop, because it starts to get beyond my skill set to do that kind of more complicated math.
Thanks, thank you. So that let's go to the next one, which is very interesting. And the question is about there are so many emerging risks, new chemical product, and you talk about PFAS. And if we go with the classical approach, it can take long time. And whether it's a responsibility of the industry, or are we equipped with better tool to understand that potential impact much quicker rather than taking the conventional route of when it will be mapped to platform like Ecotox or Comtox.
So again, an outstanding question, and I could give a long answer to that, but I'll try to keep it very shorter. So this was a big issue, right? When we first started with when the US EPA started developing all these frameworks for evaluating chemicals. You're absolutely right that you can't just be doing all this research in animal models, because it's going to take 1,000 years and new chemicals are coming in all the time. How do we run them through systems that will allow us to understand the potential for risk? And so that's where the US EPA started thinking about this issue through the TOX. It was called originally the TOX 21 program, where they were going to develop a lot of these in vitro assays to more quickly evaluate the chemicals. They now have more than 800 of these assays. A lot of these chemicals can be run through in high throughput, meaning that you can get a result fairly quickly. All of the data run for all of the chemicals so far is publicly available to everybody. It was part of the workshop I did a couple of weeks ago here. But of course, that's only targeting one endpoint at a time at either the cellular or the molecular level, which can lead to difficulties in interpreting how do you scale that up to a whole organism, which was one of the original criticisms that a lot of my colleagues had for this CompTox database, where Tox 21 turned into CompTox. I sat on a panel that looked at the comparison between CompTox and the endocrine disruptor screening program for estrogenic processes, which I didn't talk about at all today, but the US EPA developed that as well for estrogen, as well as thyroid, and for androgen. And to my happy surprise, the CompTox data from the multitude of in vitro assays pretty well matched what they were seeing in rodents for the chemicals in the environment and how they disrupted that system. So I think there's a lot of promise there. I do think further validation is important, and at least up until recently, the US EPA was working on that pretty intensively.
Good. So more questions are flowing, so we'll see, and all are very interesting. And perhaps I can also mention that Professor Propper would be around one more day. So if you want to discuss further, we can also schedule meeting, but we'll take few more for sure. There is another interesting question from Jai Lawrence that you mentioned about epigenetic changes that persist for multiple generation. And question is that if you have done something around the model organism, which we discussed today, in terms of understanding whether that changes are typically persist only few generation or it remain there indefinitely.
You know, that's also a very interesting question. And I have not dug into the literature, so I guess my short answer is I don't know how many generations it lasts. I haven't dug into that literature at that level of depth. The original study came actually under conditions of stressing moms out during the developmental process of fetuses, and then looking at their stress responses later. That was in rodents. That came out of studies in humans that had had very traumatic experiences during development or during early stages of childhood, and that has carried forward into next generations. Smoking was one of the other issues where they have seen cross-generational effects in humans. There are some studies in fish and in mice. It's hard to do because you have to get multiple generations going, and those studies are expensive because they have to go for long periods of time. I'll have to look in the fly literature. I bet there's something in fly literature that'll tell you that, but it doesn't mean if in flies it lasts this long, this many generations, that that's the same in humans or even in rodents or fish. So I guess my short answer is I don't know, but it's at least one or two generations downstream. There are data in support of that.
Yeah, and I think I can add that that question is fantastic in terms of that we have in some way scratched the surface in terms of research in these areas. We do need to run these, like what we generally say about agriculture, that long-term studies. Similar goes in this area, that we do need to run these kind of experiment for multiple generation to understand, yeah, some of the answers. Yeah, so fantastic question. Um, We'll take few more. So hang on. There is a very interesting question. Anecdotally, there seems to has been a lot of cases of women with breast cancer in agriculture region in Queensland. Is this similar in the USA?
There's definitely hot spots for all kinds of different types of cancers. A book just came out about paraquat and Parkinson's disease. So yes, I would say yes, there are hot spots around agricultural areas for all kinds of diseases. We published a paper about lymphoma, looking at pesticide use across broad swaths of the Western US, and found, and we actually started with just looking at pesticide usage and then cancers at the county level, and found that one fumigant, in fact, was much more closely linked to lymphoma, both pediatric and adult cancers with lymphoma. And with breast cancer, there's similar kinds of correlative data. We certainly know that a number of these chemicals interfere with estrogen-dependent processes or act as environmental estrogens. It's really hard to sort all of that out from things like usage of birth control early on, genetics and genetic predisposition, whether people smoked, what household products they were using, right? And so trying to figure out which chemicals might be involved in this and whether those correlations are linked to any of those individual chemicals or the broader mix of chemicals is very difficult. And again, that's where I think AI and systems approaches may help us start to sort out some of those complexities beyond the usual use of multivariate statistics that have been used in the past. And again, correlation doesn't mean causation, but you start seeing enough correlations across certain situation types, and it starts to be, you start to be able to make inferences.
All right, we are almost at the hour, but we'll perhaps try to take a last question. And Professor Propper, if you can be short with this one, but as this is a quite interesting one, that are there example for thyroid where particular chemical is toxic to animal model but not toxic to human? Or is it always what is toxic to animal is toxic to human? So I think the question is there any preferred animal model organism that better represent human for these thyroid toxicity studies?
That's such an interesting question. And all of these questions, let me first say, all of these questions have been really thoughtful, and I really appreciate how carefully people listened to the talk and understood what I was trying to convey. Is there anything that has been shown to be toxic? You look at one, if you look at anything for one animal in one study, you can't really make an inference across species to other outcomes because of those complexities of the pharmacokinetics. I would still argue that the endocrine system, our detoxification systems have all evolved to be extremely sensitive, right? You don't want to put a lot of energy, evolutionarily, you're not putting a lot of energy into insensitivity of enzymes and receptors. That said, there are differences. There's differences based on evolutionary history, as I said, of past exposure regimes. Sometimes it selects for things, so you're going to be less sensitive. So I think it could go either way, and it depends on the model you're looking at. I like using wild animals in part because of their genetic diversity, as opposed to lab animals where you have lack of genetic diversity, even say with the African clod frogs in laboratory situations, right? They've been selected for so long to be sitting in the lab, their genetic diversity is down. Humans are really diverse. And so where even some people may be more sensitive or less sensitive, which could lead to differences in potential risk for cancer. So I guess the answer to your question goes back to, I guess, my title, which is it's really complicated. The complexity of all of this with the multitude of exposures we have and the diversity we have is important. But when we start seeing constant human health impacts correlated with exposure, and we see that consistently in animal models, I think that's a red flag that needs to be addressed. I also tell people, I'm not the best person to ask what I do for a living at a cocktail party, but I also want to emphasize that for humans, we are living about 40 years past our natural lifespan, and that's because we have food, and access to medicine. And we can't ignore some of the good things that have come out of the use of chemicals. I think we just need to be careful about it and thoughtful moving forward.
No,
And
that's
again,
a
I'm past
fantastic.
time, so I'm going to stop talking.
Yeah, no, I think that's a fantastic way to kind of conclude this one about that this is a complex domain, but you really help us now in this seminar to look inside that. So on behalf of all audience, first, Professor Proper, thank you very much for
Thank
sharing
you,
your experience
everyone.
and
Thank
knowledge.
you.
And I would like to also thanks all the audience for asking these fantastic questions. Can you please click the next slide? Again,
Yeah.
this is the conclusion of the today's seminar. If you haven't already, please sign up for the coffee seminar list, and you can get the detail of the future upcoming seminars. So with that, on behalf of Professor Proper and myself, thank you very much for joining today's webinar. Thank you very
Okay.
much.